have María José Sánchez Barrena, who is the coordinator of the Group of Structure and Function of Proteins. And she is a researcher from the SEBBM, and she's going to tell us about the speakers she has invited. And today we will have 2 speakers. So thank you very much, María José.
Thank you very much, Patricia. I'm going to be fast just to give time to speakers to present their work. Our first speaker is José Manuel Pérez Cañadillas. He's principal investigator at the Protein NMR group at Instituto de Química-Física Blas Cabrera, and he will present the work with title Viral Nucleocapsids as Potential Targets for the Discovery of New Broad-Spectrum Antivirals. Thank you, Jose, and the floor is yours.
Why viral nucleocapsids as potential target for discovery of new broad antivirals? Okay, if we look into the sample of SARS-CoV-2 nucleocapsid in general and the organization of the genome of this viral, the viral has 5 structural proteins. Nucleocapsid is one of these proteins. And if you look into some detail of the nucleocapsids, you can find, or I did the numbers myself, maybe they're wrong. They are big numbers, difficult to compare. If we think about a virus of a diameter of 100 nanometers, and between 700 and 2,000 molecules of NCAP for each virus. These are numbers I collected from the literature. You will expect very high concentration of this protein inside the particle, viral particle. Actually, if you make these numbers into volumes, they can occupy between 10 and roughly 10 and 25% of the volume of the virus, which is quite a lot. So we will expect any disruption of the interaction between the RNA and the nucleocapsid, or between the nucleocapsid protein itself, will cause probably a disruption in the assembly of the viral particle. And this will hopefully lead to a malfunction in the assembly of the of the virus, and at the end cause difficulties in the propagation of the viral cycle in general. Okay, this is one of the reasons why we choose nucleocapsid. The other reasons we choose nucleocapsids as a target, one of them is very prosaic. We are a very small group, and given the jungle of groups working on COVID at the time of the pandemic, most of them we thought they're going to target other more attractive targets like the enzymes of the virus, let's say the RNA-dependent RNA polymerases, or the protease of the virus that are targets which are often used for antiviral therapy in viruses. We have many examples of the HIV and other viruses. And the nucleocapsids is not so often choose as a potential target. So we think it out of the box to be more competitive against other groups or companies leading for different more common or usual strategies. There are also some reasons to choose nucleocapsids as well, more other reasons. One of them then is that it's very highly conserved protein. It's one of the proteins that changed the less along the pandemic, especially in particular within the folded domain. The protein is a 400 amino acid protein, more or less, and it has 2 folded domains, an N-terminal domain, which behaves as monomeric, and a C-terminal domain that makes a homodimer. It's also a highly abundant protein, as you see from the numbers figures I saw before. And as well as in the packaging, it also participates in different cycles during the replication and transcription of the viral genome. So it's important for the viral cycle as well, apart from the packaging, apart from its structural role. Okay. So the strategy we follow is basically to use NMR to screen libraries. We started with a fluor-19 library, and we find some hits. The fluor-19 library comes from the CNIO, collaboration with the CNIO. We screen different compounds, about 250 compounds, and find about half a dozen of hits. And these half a dozen of hits, we try to identify common structural features, get their interaction, better characterize their interaction by using NMR, and also other structural techniques such as X-ray crystallography or biophysics. And once we have these hits, we tested them for in vivo inhibition in collaboration with the group of Margarita Sainz at the CBMSO. The collaboration with the X-ray crystallography group here at the Blas Cabrera Institute with Beatriz González was essential also for the work. Once we get this information, we end up with some structural data. We use this data to simulate by computational docking what will be the interaction with similar molecules. And the final goal to proceed in the future with the project is to find a medicinal chemistry group which will be potentially interested in development and evolve the proteins, the hits we found during the process of the study. Okay, this is a brief introduction about why using NMR for protein ligand interactions to characterize protein ligand interaction. And the main reason is because we can have the best of both worlds. We can use ligand-directed methods to identify from a mixture of compounds which of them interact selectively with a protein, or we can look into the protein to identify where the binding site spots are. I will exemplify this with the case of nucleocapsid protein. First, we start with the fluor-19 library. Basically, the process is looking at the fluor-19 NMR spectra of mixtures, typically 8, 9, 10 compounds each. Typically, they have one or two signals per compound, so they are spectra which are easy to interpret. And we titrate the protein, in this case, full-length NCAP from from SARS-CoV-2, and look for those signals to suffer a decrease in their intensity. The fluor signal is very sensitive to the transient interactions with the protein, with big molecules, and some of the features of the big molecule are transferred to the signal, and so they are easy to spot in the experiment. We identify, as I say, less than half a dozen of hits, but two of them contain this structural moiety, this chemical moiety. It's a benzothiazole moiety, it's an heterocycle. So given that we have this common moiety, we thought that maybe we can find and look for other benzothiazole that we can buy from suppliers. And we buy a number of benzothiazoles from different suppliers and tested them, for instance, using mixture of them, using, in this case, STD experiment. STD experiments, saturation transfer difference experiments, stands for this. These are the name of the experiment. Basically, what we did is we selectively excitate the signals of the protein and transfer some of the energy selectively to those compounds which transiently interact with the protein. We did a reference experiment and an on-resonant experiment with the excitation on the protein on, and performed a different experiment to spot those compounds interacting. Rather than giving only the information of whether it interacts or not, also give information of whether these sites, the signals which are suffering from the STD effect, are close or far from the protein. And this is something so we can get some quantity information. Among the different benzothiazole we assay, there is one which is riluzole, which is an LAS approved drug. Riluzole is a drug been around for almost, around 30 years now. It's been the first drug and one of the few drugs being approved for treatment of amyotrophic lateral sclerosis. It's a well-known drug, and so far this is the only application. There are other possible applications under research, all of them related to mental, neurological disorders or diseases. But so far as I know, there are no applications as a possible antiviral. Okay. Once we have the benzotiazole compounds proof that they are binders of the nucleocapsid, we inquire the system now where actually the protein binds. So for that purpose, we'll switch look into the protein spectra now. In this case, the CTD NMR spectra. This is an HSQC spectra which identify basically positions along the sequence of the CTD domain, which is, as I remember is an heterodimer. In the picture here, we saw the effect that caused titrating benzotiazole, just the one plain one, with on the protein signals. The reference spectra is the black signals. Red signals are those in the presence of the compound. We can basically map these signals, and quantify the effect. And in this case, we did that for all of the compounds we tested and identify for most of them a clear spot nearly around the amino acid 338, or the NH of the 338 amino acid, which is a lysine in the sequence. And because we have the three-dimensional structure of the protein, we can actually more or less well define the binding pocket of this family of compounds. We also this binding pocket coincides with the nucleotide binding pocket. This nucleotide binding pocket has been identified in a work from Marçal Villar group in Valencia. And we also quantify the affinity, the apparent affinity of the different compounds compared with the GTP. And some of them has similar affinity or slightly lower, weaker. We have one which appears to be better affinity than the GTP. Okay. We try to get crystal structure of all the compounds, but we manage only to obtain two crystal structures of complexes, protein ligand complexes. In this case, in the Ridusol, all of them are being, in both cases, were obtained by soaking. We try co-crystallization, but unfortunately, we didn't have luck in getting co-crystals. When we see this structure, we found that the Riluzole, for instance, rather than mapping the site that has been identified by NMR, maps a crystallographic interface between the two of the homodimers. Obviously, we don't know whether this crystallographic interface has a biological relevance or not, but consider that in the biological context, the concentration of the protein is so high, we may thought that this may be the case in the biological, in the real world as well, but we don't have so far evidence for that. In the case of the 608 benzotiazole, the compound actually mapped both information coming from the NMR and X-ray crystallography are coincident. These are some details about the binding pockets. You can see some stacking interactions across the interface. In the case of Riluzole, with the aromatic stacking to the tyrosine, some specific hydrogen bonds with water molecules, and the 3-fluoromethyl groups stacking to the tryptophan. The binding pocket, the nucleotide binding pocket, is where the other compound binds to the left here. Okay, here to the left, to the right, sorry. And you can see there are specific interactions with water molecules, structural water molecule with the 2OH moiety. The NH that we see more shifted in the chemical shift data is basically the NH of this lysine here. Sorry, I cannot. Yeah. So this lysine here, which is actually the 3,3A lysine. And this information is very robust with the data we have from the NMR. Okay. As I say, the binding site is the nucleotide binding site. We can compare the structure, our structure, with the structure of Marshall group, and you can see that both aromatic rings are more or less on the same plane. They make stacking interactions with this tryptophan, T-stacking, but the GTP is a bigger, larger molecule and has contact with the sugar and the triphosphate group with other parts of the molecule. In collaboration with the group of Margarita Sainz at SEBBM, they perform the antiviral activity, testing both human respiratory cells infected with either SARS-CoV-2 or another coronavirus, human coronavirus, this one, the one on the right, which is from a different family of coronaviruses, but structurally related. In their studies, they assay the virus titer to test for the concentration to inhibition concentration, but also their toxicity of the compounds. You can see that the riluzole has a relatively weak binding affinity, but when you make the selectivity index, taking into account also the toxicity of the compound, we have a figure which is about 10, which is, as far as I know, considered to be a good figure to consider a drug as a potential, a compound as a potential drug candidate. We see some similar effect, but with lower affinity for the case of the other coronavirus. So maybe these kind of molecules are also has some broad activity against other coronaviruses. To our surprise, in the case of the other compounds that apparently has better affinity, we didn't see any effect on the any antiviral effect. Okay, so now knowing that the benzodiazole family of compounds are capable to interact with nucleocapsid protein, we think in using the structural information, the rich structural information we have, to make models about other different compounds related to this benzodiazole. For this purpose, what we did is to construct a in silico library of benzodiazole, all of them below, let's say 300. I think it was around 300 molecular weight, so a small compound in all these cases. All of them extracted from Zinc 22 or PubMed chemical libraries. We use some restrictions from the X-ray structure we have, and we also include structural water. This is a feature that is possible using the gold docking program. And these are some of the statistics of these docking experiments we perform. In the statistics, basically, we compare the fitness, which is an internal parameter of goodness of the docking. Also, RMSD to the reference X-ray structure, and some other values regarding the energies of each bond or barrier terms. The dots, red dots, correspond to the redocking of the T replica of the benzodiazole OH, which is the same as in the crystal structure. You can see that these dots are very good energy values, which is a very good solution among the ones we tested. So we are very close with this towards the best option, the best possibility. Okay, so the goal with this docking was to get whether we can expand. This is a movie showing all the docking structures we obtain. Is to obtain whether they are adjacent site to the nucleotide binding site that can be exploiting for a specific recognition. This is what is tended to see in this summary figure on the right, where we can see different substituents at the different position of the benzodiazole ring, and whether they are capable to interact with the specific side chains of the protein. This is complex figure, but we can map this basically this binding accessible site on the structure of the protein, and you can see that it's expected, it could be that we can extend the binding site toward other green areas next to the binding site, the primary binding site, to the left or up and down of the current binding site. So we are happy, and we think we can develop or improve the binding affinity and structurally of this binding site. There are still a margin to exploit. Okay, so far these are these are the results of what we have published. Obviously, what we want to aim at the long term is to find molecules that has broad activity against other coronavirus, and this is a really outdated picture of what we know in terms of sequence of coronavirus sequences in 1 year ago. Probably these figures raised a lot, but still we have 1,000 of sequences from different families of coronavirus. The ideal situation will be to find molecules which are capable to interact with more or less universally. So how we can get universal binders for coronavirus nucleocapsids? Perhaps a strategy to anticipate to a potential SARS-3 pandemic. Okay, we think about this strategy, which use a combination of the things we learned from SARS-CoV-2 using fragment libraries to identify common binders with other nucleocapsids, in order with the goal to obtain a chemical library, either for in silico screening or even for actual experimental screening of new nucleocapsids. And we can also exploit, I think it's valuable to exploit all the information we have with the database using artificial intelligence methods to calculate three-dimensional structures to get to populate a nucleocapsid selected structural database. So we can combine both the best of the two worlds in silico binding experiments, or even in actual experiments, in experimental experiments. Okay, we have done some take some steps towards this goal. First step is to characterize in the institute, in my group, a small chemical library of fragments. It's about the size of the chemical library we use in the CNIO, but this time we use STD as a screening method. And we test this library against the SARS-2 NCAP and also against MERS NCAP, which are the two more pathogenic coronaviruses that have appeared in the last decade. To our happiness, we found compounds which are present in both molecules, so we can expect there will be, hopefully, molecules that are capable to interact with more broadly with nucleocapsid protein. This is something we need to test and to explore. On the other hand, we can use and exploit the information on the database, and thinking about what could be the next potential bad coronavirus to cause a pandemic. One place could be on bad coronaviruses. There is a lot of bad coronaviruses having been sequences. This is the one. This is just a comparison about the structural sequence diversity that you have against these common coronaviruses just from bats. From different families, just focusing on the CTD domain. This is just a heat map to give you an insight about how are these different in terms of sequences, but they are not so different in terms of the structure. We compute the structure of more than 20 or 30, I can't remember the exact numbers of nucleocapsids using AlphaFold at CESGA and Galicia Supercomputing Center. And as you can see, in all the cases, the nucleotide binding site appears to be conserved, at least the location of the site. We don't know whether this could be a site that could be exploitable for this targeting new antivirals. Okay, so far this is the end of my talk. I'd just like to finish thanking other people, both in the NMR and X-ray group working on the project. Especially thanks to María Ángeles, who was the postdoc doing all the studies in the X-ray studies, has a lot of work on that, and Beatriz González as well for the fruitful collaboration with her. I want to thank also to Lourdes Infantes, which is a specialist in the GOLD program. She is a specialist also in the chemistry database, and is the person I know knows the most about these tools. From the CBMSO, from Patricia, Margarita, and also to Paco Sobrino and Encarna for the fruitful comments. Those are the guys that really knows about the biology of the virus, and I learned a lot from them, from talking with them, and also to the CENIO Biophysics unit that give us access to the library. And all of you for your attention. Thank you very much. Okay.
Thank you very much, Jose, for the interesting talk. I'm a bit concerned about people listening to this talk in YouTube. So if there is any problem, please, could you please report through the Zoom link, please? So it is now time for questions. Just for I will start until people start to get warm. So Jose, how directed the chemical library at CNAO is? How chemically diverse is it? Could you maybe think about looking for, very different things, or you were thinking about targeting the nucleotide binding site and that library is specific,
Well, as far as I know, they are not designed to target to be nucleotide mimetics, for instance. It's a library from I think from a commercial company. I don't have the details, but it's a fragment library, so it's made of small compounds. Obviously, you cannot cover a huge chemical diversity with just 250 compounds, but it's wide enough just to get a compromise between the speed of the process of screening by using a medium throughput technique as NMR, and having a reasonable number of chemical structures just to get some hits. Obviously, the same happens from the other commercial library we had here at our place. So yeah. So they are basically small compounds. I think all of them below 300. So they are not intended to be drugs. It's just fragments.
So anybody
you will combine both.
So anybody thinking about doing drug discovery and trying to look for hits, you would recommend that library as a starting point?
It's a starting point, yeah. I think what is being recommended for fragment libraries is slightly larger. So it's in the range of 1,000 to 2,000 to use by NMR, I mean. There are other techniques which has higher throughput, and then you can target those. And there are other experiments to do high throughput, not using biophysical methods, for instance, like NMR or using just enzymatic assay methods. It depends really on what you can screen on your target molecule. In the case of, for instance, things like the someone has stopped my video. I don't know whether you can hear me. Can you hear me? Yeah. Okay. Yeah. So basically, if you are targeting, for instance, an enzyme, you can look for loss of enzymatic activity. So if you have a good enzymatic assay, That's the way forward, probably. In the case of nucleocapsid, it's difficult because you don't have an enzymatic assay, so you need to look for things that affect its, don't know, stability or binding ability, or just looking direct to the signals of the nucleocapsid is what we're doing now with NMR. We're looking actually to the effects on the ligands of the target molecule.
Okay.
Okay, I just have one question. Well, I'm totally out from this field of research, but I think it's really interesting the way you approach to find a drug that could be valuable. So you were just based on the structure, not on the function? Like that drug that also works for Ella, it's a glutamate released drug, right? But the thing is that it's more related with the structure of the drug, but not with the function of the drug, the way you find an effect, right?
Yeah, well, obviously we don't have an idea about what is the mechanism of action of the drug in this case. The way we fish does the drug is by using a screen protocol. We don't really can tell whether this drug actually performs his action by interacting with the protein itself, or it has a different mechanism. We really don't know, to be honest. This requires further investigation. What we see, what we identify is that there are ways to using this family of drugs, the benzothiazoles, as mimicking the nucleotide binding site, which is good. It will be other compounds, obviously, targeting that binding site. But obviously, we can. This is true that we don't approach using a function, and the reason behind that is because we don't have. Other people may have it, a good functional experiment to test that. There are functional experiments. If you look at, for instance, the capability of the protein to form biological condensate, we can screen whether the drugs interfere with the function of forming biological condensates, which meant to be important for the function of the protein if we look up this function. Or we can have an assay that tell us something about the capability to interact with RNAs, for instance, but it's something we need to decide.
Okay, thank you.
Okay, so thank you very much, Jose. I think that, yeah, for a question of time, we will move to the next speaker.
Thank you very much.
Thank you very much. It was very interesting. I would like to remind the people that are connected that we have had problems with the YouTube link, and although the talk is being recorded and will be uploaded in our YouTube site, those that want to listen properly to Maria, I would suggest to go better through the Zoom link, please. So our next speaker is María Lucas. She's a investigator, principal researcher del Instituto de Biomedicina y Biotecnología de Cantabria, a mixed center between Universidad de Cantabria and CSIC. And she's going to present some of the work that is running right now in her lab with title Decoding the Logistics of the Endosomal Network, Structural Insights into Integral Membrane Protein Transport and Recycling. Thank you, Maria, for accepting the invitation, and the floor is yours.
So good evening, everyone. Can you hear me well? Yes? Okay. So first, I want to thank the SEBBM and Maria Jose for the invitation and this amazing opportunity to share with you the research my group has done in the last years. So when we look at images of eukaryotic cells, we see that they are filled with membrane and different organelles, and those of us who work in intercellular trafficking, we're interested in knowing how molecules, proteins, move between these membranes and organelles. So what we want to know is what are the heavy vehicles that transport the cargo, which routes do they use, which cargo is transported along its route, and how do the vehicles read the barcodes that are present on the cargo. So that's why we can consider that intracellular trafficking is like the logistics of moving molecules between compartments inside the cell. So if there are errors in these trafficking pathways, then proteins will accumulate in the ground compartment, like you see here with this accumulation of boxes, and this is related to diseases, including neurodegenerative disorders such as Alzheimer's and Parkinson. So these diseases affect neurons, and they need a highly efficient intercellular trafficking system because the vesicles they have to need to travel long distances. So these trafficking routes are also exploited by intercellular pathogens such as bacteria and viruses, and which hijack them to survive and move within the cell. So the cargo that we study are integral membrane proteins. So these proteins are essential for maintaining cellular homeostasis. So they, for example, mediate responses to external. So okay, I think it's here where it crashed my system. Sorry. So I was telling you that vesicle trafficking involves 4 main steps that this happy vesicle illustrates. So first is the budding, called proteins are recruited to the donor membrane to induce vesicle formation and to select the correct cargo. Second is the movement. The vesicle travels along these cytoskeletal tracks with the help of molecular motors. Third is the tethering. The vesicle establishes the first physical link with the target membrane thanks to these long tethering complexes. And the fourth is the fusion. The D snares present in the vesicle and the T snare present in the target compartment, they form a 4-helix bundle that allow the merging of the membranes and the delivery of the cargo to the target compartment. So all these steps, our research focuses on the first one, and that is how these code proteins, they can select the correct cargo and they form the membrane to produce the vesicular carriers. So and the routes that we investigate are the endosomal recycling pathway. So integral membrane proteins located on the plasma membrane, and they reach the endosomes by endocytosis. And endosomes, they work as a sorting compartment, and the fate of these proteins will be so no, it's okay. The fate of these proteins will be okay. See, the fate of these proteins will be sorry. I am okay. It's I'm confused. I'm sharing well my.
Maria, now your presentation is closed.
Okay. Yeah.
But before it was okay.
Before it was okay. Okay. Sorry. Okay, I was telling you that endosomes act as sorting compartments, and here the fate of these cargo proteins, of these integral membrane proteins, will be decided. So they can follow a degradative pathway or a recycling pathway. So these, so there are some proteins that are ubiquitinated, and the SCORE complexes recognize these proteins, and they are internalized in intraluminal vesicles. And the late these endosomes, they mature to late endosomes and fuse with lysosomes, and these proteins will be degraded. And opposite to this degradation pathway, there is a recycling pathway. And there are several complexes that are involved in this recycling, and the integral membrane proteins are can be then transported back to the plasma membrane or to other organelles where they can be reused, and they can also modulate the functionality of these organelles. So there are several recycling complexes. So, for example, retromer combined with SNIX27 that mediates the transport of cargo from endosomes, early endosomes, to the plasma membrane. So on retromer, when it's combined to SNIX3, it will transport this cargo to the Golgi network. And so this route is also mediated by escape-1 complex that also is has been suggested to assess this sorting mixing 27 retromer pathway. And some years ago, it was also discovered a retriever complex that interacts with SNIX17 to recycle cargoes from early endosomes to the plasma membrane. So this complex also interacts with the CCC complex and the WAS complex that is involved in the polymerization of actin. So it is known that the retromer complex and the escape complex, they produce this recycling, deforming the membranes and producing these tubular vesicles that differ from the classical round vesicles, like the ones produced by clathrin. And so you can see here in this video how the so these tubular vesicles are produced. So here is one retromer subunit has been labeled with GFP, and you see these tubules that are emerging from the endosomes. So I have done some research with two different recycling pathways, the one that is mediated by retromer and SNIX3, and the one with retriever and SNIX17, so that I will present today. So first, so when we studied this retromer SNIX3 pathway, what we wanted to know was how is the cargo specifically selected and loaded into vesicular tubules, which is the structure of the code formed by, and what is the structure of the code formed by retromer. So this study was published in Cell, and I want first to acknowledge the authors. So I did this work when I was as a postdoc in Aitor Hierro's lab, and we collaborated with Juan Bonifacino's lab in the NIH in Bethesda. So the retromer complex is composed of three subunits, BPS26, BPS35, and BPS29. And so we crystallize and solve the structure of the complex between BPS26, the N-terminal part of BPS35, and SNIX3. And we also included, so in the crystallization, the cytosolic part of this integral membrane protein, this DMT1 protein that contains consensus sorting signal. So this is the structure that we obtained. We could describe the different interaction surfaces, like the interaction between BPS26 and BPS35, how SNCC3 interacts with both subunits of the retromer complex. And interestingly, we saw that the cargo peptide is interacting with both BPS26 and SNCC3. So, and this shows that sorting exin 3 is not only recruiting retromer to the membrane, because this is a protein that has a PX domain that binds phosphatidylinositol 3-phosphate present in endosome, but also acts as a adapter protein for selecting the cargo. So we did different ITCs with different compositions of proteins, and what we saw is that DMT1 recognition involves binding to both retromer and SNCC3, and that retromer binds SNCC3 only in the presence of cargo. So we model we produce a model of the full-length retromer bound to a transmembrane protein on membranes. So what our results indicate is that the recruitment of retromer to the membrane is coupled with cargo selection. So we wanted also to study how this retromer complex produces a coat and deforms the membranes to produce these tubules. And so this video summarizes what we did. So first, this is the overall structure of the complex that we crystallized. So this BPS26, BPS35, SNCC3, and the cargo DMT1. So this recycling motif has a central leucine that is in a hydrophobic pocket that is formed in BPS26, and we observed that this cargo interacts with both BPS26 and SNCC3. So it was known the structure of the other part that we didn't crystallize, so the last part of BPS35 and BPS29. So we did some SACs and we could model the full-length structure of retromer and also the dimer. Okay, and so since we had the complex with the SNCC3, so we could model how this dimer interacts with the membrane. So it forms these arch-type structures, and we also observed that BPS26 has some positively regions that can interact with the membrane. So this PX domain of SNCC3 is also present in other sorting exins that have also bar domains. And so we align these proteins, and then we see that they are from parallel configurations that we can repeat so many times that we want. And when we apply the tip-to-tip contacts between these bar domains, it generates this nice helical organization of the sorting exins and retromer around the membrane. Okay, so we propose this helical organization with these arcs of dimers of retromer around the membrane, and so some articles that were published later with cryo-EM structures, they confirmed that this helical organization of the retromer code. Okay, now I will move on to the work that we have done with the retriever complex. So, as I said, retriever, along with SNCC17, recycles integral membrane proteins from early endosomes to the plasma membrane. So the work I'm about to present was published recently in EMBO reports. So you can see here the link if someone is interested in learning more about this work. So I want first to acknowledge the authors, especially the talented PhD students in my lab, Aurora Martin and Ivan Mendez, and our collaborators in the IFTEC Institute, Juan Carlos Acosta, and David Alvesa in the Biophysica Institute in Bilbao. So the questions that we wanted to answer in this project were: how do retriever subunits assemble to form a complex? Is the interaction between retriever and SNCC17 regulated by cargo recognition? Which is the structural organization of the retriever SNCC17 complex? Is there an optimization mechanism for retriever binding in the absence of cargo? And how is retriever recruited to membranes? So our first question was, how do retriever subunits assemble to form a complex? So retriever is composed of 3 subunits, BPS29, BPS35L, and BPS26C. So this complex shares with the retromer complex the BPS29 subunit, and structural prediction of the other subunit indicate that there are some structural homologies between the retriever and retromer complex, although the sequence conservation is low. So, but it was not known how these subunits interact and the structure of the complex. So we first overexpress the retriever complex in insect cells and purify the complex as a stable heterotrimer. And we also purify the individual proteins BPS26C and BPS29. So we tried with different approaches and constructs to purify BPS35L, but we didn't get any soluble protein, and we think it's because this protein is unstable in the absence of the other subunits. So to investigate the possible potential interactions between BPS26C and BPS29, we did gel filtration chromatography, but these proteins, they didn't coagulate, so they don't interact. So this indicates that BPS35L serves as a core protein for the assembly of the other subunits. So to map the interaction domains with the other subunits, we generated several deletion constructs and purified the resulting retriever complexes. So we found that BPS26C interacts with the N-terminal region of BPS35L, here highlighted in red. And interestingly, BPS29 shows a dual interaction with BPS35L, since it requires for binding both the N-terminal and the C-terminal regions highlighted here in blue. So to validate this structure, we use AlphaFold to model the structure of the complex. And this model aligns with our results. So we observe that BPS26C here in red is interacting with our predicted region, and that BPS29 interacts with both the C-terminal region and this extended N-terminal region of BPS35L that produce an intramolecular interaction between the N-terminus and the C-terminal part. So to further validate this model, we use SEXAC, so size exclusion chromatography coupled to small angle X-ray scattering. And we, so the SAX envelope that we obtain, it aligns well with our models. So confirming that the retriever model represents with a high accuracy the conformation of retriever in solution. So interestingly, this model shows that retriever adopts a distinct conformation compared to retromer, despite their structural homology. So retriever is a more compact molecule with greater curvature, while retromer displays a more elongated structure. So 3 articles have been published recently that present the cryo-EM structure of retriever. So our predicted model aligns well with this structure. and only slight differences are found. So next, we investigated if the interaction between retriever and SNCC17 is regulated by cargo recognition, as we have seen before in retromer. So we first analyzed the binding affinity between SNCC17 and several known cargos of the sortinexin. So sortinexin 17 is composed of 3 domains: a PX domain that interacts specifically with the phosphatidylinosyl 3-phosphate present in early endosomes. Then a firm domain that interacts with the cytosolic tails of the transmembrane proteins that will be transported, so the cargo. And these proteins, they have to have a special sorting motif, that is this MPXY motif. And it has also a C-terminal domain, which structure was predicted to be disordered. So we have worked with these cargos, this LRP1, APP, the amyloid precursor protein, integrin beta-1, and L2, that is the L2 capsid protein for human papillomavirus type 16. So to measure the binding affinities, we did fluorescence anisotropy assays with several peptides covering this recycling motif and labeled with a fluorophore. And we found that LRP1 and L2, they both have a high affinity with a KD in the low micromolar range, while APP and integrin beta-1 has a lower affinity for sortinexin 17. So to analyze the effect of the cargo in the interaction between retriever and SNCC17, we did some pull-down assays using MVP SNCC17 immobilized on amylose beads, and we incubated first this protein with the cargos and then with retriever. So we observed that in the absence of cargo, there is only a modest retention of retriever, but when we added the cargos LRP1 and L2, there is a significant enhancement of these binding of retriever to sortinexin 17. So with that the cargo triggers this retriever recruitment. And so in contrast, APP and integrin beta-1 didn't show this a significant enhancement, likely because of their low binding affinity. So we observed that retriever is directly interacts with SNCC17 and that the cargo binding to SNCC17 enhanced retriever recruitment. So next we examine the structural organization of the retriever SNCC17 complex to identify the key interaction determinants. So using AlphaFold, we model several retriever SNCC17 configurations, and in all models, the C-terminal 18 residues, they interact with a groove in the interface between the BPS26C and BPS35L subunits. So to validate this complex configuration, so we perform fluorescence anisotropy assays using a peptide of the last 18 residues of SNCC17, and we observe that retriever binds to this peptide with a low with a KDO in the low micromolar range. So if we use a complex with only BPS26C and the N-terminal region of BPS35L, we obtain the same affinity, but in contrast, BPS26C alone or BPS29 didn't bind or the complex of BPS35L and BPS29, so only a low binding affinity. So these results confirm that the C-terminal region of SNCC17 binds at the BPS35L BPS26C interface. So, and we then studied the key residues involved in these interactions. We observed that so that the last residue of SNCC17, leucine 470, forms a hydrophobic interaction with the tryptophan, and the carboxyl group is forming some salt bridges with this arginine. So this interaction is also stabilized with by this hairpin of BPS35L that produces some salt bridges with the SNCC17. So we validated these predicted interactions experimentally. So first with atrucation of the last 4 residues of SNCC17 and the substitution of the last leucine with glycine. And we observed that these mutations abolished retriever binding. And when we mutated this arginine and tryptophan, we didn't see any binding, and this lysine and arginine of the hinge region, the binding decreased 5-fold. So these results demonstrate that the C-terminal leucine from SNCC17 is essential for retriever binding, and that the BPS35L pocket accommodating the C-terminal leucine of SNCC17 and the hinge domain are both critical for binding. So we had observed before that the cargo binding to SNCC17 enhanced retriever recruitment. So therefore, we asked whether there is an autoinhibition mechanism for retriever binding in the absence of cargo. So the published 3D structures of SNCC17 include the PX and FERN domain bound to the cargo, but there was no structure that includes the C-terminal region. So that's why we use AlphaFold, and we obtained the structure of this region, and we saw that this beta strand followed by this alpha helix occupies the cargo binding pocket. So, and when we had a look at this sequence, we observed that it's a cargo mimicking motif, but it has no, because there is this motif NXXF that is structurally similar to the recycling motif for proteins that bind firm domains, that is NPXY. So then we wanted to know what happens when SNCC17 binds the cargo, and we see that this C-terminal domain is replaced by the cargo, and this produces a shift and a big conformational change in this C-terminal domain of SNCC17. So we have proposed this model. So in the absence of cargo, the cargo mimicking region interacts with the cargo binding pocket, and this leaves the retriever binding region poorly accessible, and SNCC17 cannot recruit retriever. But upon cargo binding, this region is released, and now this retriever binding region can act as a hook to engage the retriever complex. So to validate this mechanism, we introduced mutations in SNCC17 to restrict the autoinhibited conformation. So we mutated this tryptophan, valine, and histidine, and we observed that in the absence of cargo, there is an enhancement in the interaction with retriever. So this suggests that we have disrupted this autoinhibitory interaction the C-terminal part is released, and then that's why we don't need the cargo for the activation of this binding. So we mutated also these phenylalanine and asparagine that are involved in the autoinhibition, but we don't see binding to retriever. But when we had a look at the structure, we observed that these residues are also interacting with retriever. So we think that the autoinhibitory conformation has been released, but since these residues are key for the interaction, we don't see the formation of this complex. And so these results support the proposed autoinhibitory inhibition state for retriever association that is unlocked by cargo binding. So finally, we asked how is retriever recruited to membranes. So to address this, we perform membrane binding studies using groups, unilamellar vesicles. So when we incubated these vesicles with GFP SNCC17, we observed that the protein is bound over the entire surface of the group, but when we incubated it with retriever, we didn't see any binding. But however, so when we are incubating both proteins with the groups, we see that both bind to these membranes. So that's so we proposed this model for membrane, so that membrane associations induce a conformational change in SNCC17 that is similar to the one that we observe with cargo binding, that allows that the C-terminal region interacts with retriever and captures retriever. And this could also facilitate the cargo selection. So to quantify the membrane association, we also performed liposome co-sedimentation assays with liposomes containing phosphatidylinositol 3-phosphate. So as observed with the groups experiments, we saw that SNCC17 bound strongly to liposomes. Retriever alone did not sediment with liposome, but in the presence of SNCC17, retriever sedimentation increased significantly. And interestingly, adding the LRP1 cargo resulted in a modest but significant further increase in retriever recruitment. So, and as expected, liposomes lacking phosphatidylinositol 3-phosphate failed to recruit SNCC17 and retriever. So these results demonstrate that SNCC17 binding to phosphatidylinositol 3-phosphate containing membranes facilitates retriever binding to SNCC17 in the absence of cargo, and with cargo potentially providing additional enhancement. So we have proposed the following model for retriever recruitment to membranes. So sorting exon 17, it has an autoinhibitory conformation that when the C-terminal part is bound to the cargo binding pocket, that leaves this sorting exon in an off state and prevents the assembling of the recycling machinery. But when this sorting exon interacts with the cargo or with the specific membranes that contain phosphatidylinositol 3-phosphate, it turns this sorting exon in an on state with this C-terminal region release, and it can recruit the retriever complex, and then the recycling can occur. And so we suggest that integrating these two recruitment mechanisms ensures an efficient simultaneous membrane coating and cargo selection. So going back to the parallelism between intercellular trafficking and transport logistics, So I hope that today you have learned that retriever and retromer act as recycling vehicles, that they recycle integral membrane proteins, and that structural studies are great tools to know how the barcodes, the recycling signals of proteins, are recognized. So, and to finish, I would like to introduce my current team. So I co-direct the Design and Protein Engineering Lab with Gabriel Moncalian at the IQF-CSIC Institute in Santander. So I will also like to acknowledge the funding that has supported our work in recent years. And so we are currently looking for new PhD students to join the lab. So if anyone is interested, please feel free to contact us. And thank you for your attention. And so I'm sorry for the the technical problems that occur in between. So, okay, we'll.
Okay, thank you very much, Maria.
Yeah.
Is there any question from the audience? I can start if you like. So, thinking about neural disease and the importance of having active proteins, integral proteins on the membrane, or out of the membrane, how conserved are these recycling motifs? I mean, would you, because you are talking about recycling motifs of maybe 3 amino acids, but you are using longer peptides, which are not conserved in those regions. Would you be able to think about those protein-protein interaction interfaces as a target place, to drag and try to control the presence or absence of a certain protein, membrane receptor, important for any neural disease? How do you see this?
Yes. So it's true that these recycling signals, they are present in a lot of different cargos. So then to inhibit these pathways, it will affect a lot of several cell processes that are essential. So, but for example, what it has been demonstrated is that to have a retromer that is more stable, it helps treating some models of Alzheimer's disease. They have done it with mice and in cells. And so what they have developed is some chaperones that stabilize the retromer complex. So these some chemical chaperones. And then so you increase the recycling process, then you decrease the bad effects that happens when this recycling is not functional. So, but because, for example, it's known that APP, the amyloid precursor protein, it follows an amyloidogenic or non-amyloidogenic pathway. So, and when these recycling pathways are not working properly and the APP is stuck in the endosomes, then the amount of amyloid peptide is increased. So that's why it's very important that these recycling pathways are working properly. I don't know if I answered your question.
Yeah.
So it's more like to stabilize and to increase these pathways and not to inhibit these processes.
I think Jose Maria has the hand.
Okay, can I thank you, Maria, for your talk. It was really interesting.
Thank you.
And congratulations for your work. I have a curiosity, maybe miss some from your talk, but towards the end part of your talk, you describe a C-terminal part of the complex that works as a target mimicking molecule, no? That the target mimics somehow the binding of the target. Since this part of the molecule is on the molecule itself, it has some advantage to bind towards, to perform this mimicking effect, no? So I wonder whether this binding is so suboptimal in terms of affinity compared to the binding of the to the cargo.
Yes. Yes, so we measure the binding affinity. So I have not shown this, but we saw some binding. So it's lower than the cargo, but there is this because of this. So we prove this intramolecular interaction.
Yeah.
Not because we have only the alpha structures and no crystal structure, but we prove it also. Not taking only these 18 residues and looking at the interaction with the protein. So it has to displace its own intramolecular interaction, but we can see some binding. And also we did this with interaction studies with the mutants that were designed based on the structure. And we also observed that then it is affect this binding. So we proved that this intramolecular interaction is occurring.
Yeah, I have no doubt about that. I
Sí,
thought that that maybe for the molecule itself, it will be good to have to be basically suboptimal binding. Otherwise, it will block
yes,
more permanently
sí.
the
Yes. No, it's
capacity to
not.
bind the
So
target.
then the cargos they bind with a higher affinity. So
That's
it's and it's also we see also some binding in the absence of cargo. So and then we think there is also some relaxation so that not all the molecules and so that there is some movement there and that the retriever binding domain is at some point also exposed and can interact with retriever. But we see is that so there is an activation when it binds to cargos and to the membranes, and we think that these interactions they so the one with the cargo is because it displays because it has a more affinity for this pocket, and then it displays this autoinhibitory conformation, and the one with the membrane. So we propose that it's a similar one, no? That the interaction with the membrane, also because the PX domain interacts with fatty inositol 3-phosphate on the membrane, and these interactions produce some changes that release the C-terminal part. So we try to do some molecular dynamics and model this, but so we see some movement, but we couldn't observe the total release of the C-terminal part, but maybe you have to do some longer simulations.
I have a question, not so related, but yes, because for example, with the endosomal recycling pathway is something typical of lipoprotein metabolism, and that's something we work with. And this is the case for the LDL receptor, and I don't know if you're familiar with PCSK9. Which works like it links to the LDL receptor, which is endocytosed, and then it targets the LDL receptor towards the degradation so that it won't have the recycling. So my question is, all these processes that you are studying are related how with this? I have shown that you have studied the LRP1, of course.
Well,
yeah.
Yes. So there are some articles that they saw, no, that if for example, some of the retriever subunits are mutated, there is some effects in hypercholesterolemia. So because all these lipoproteins receptors, so some of them are recycled by the retriever complex. So the recycling pathways are also very important so that this lipoprotein metabolism is efficient.
Okay.
Okay, so I think that We can't finish right now because we are having a huge delay, and we want people to also finish their experiments, I would say. So thank you very much to our speakers, and also to the audience for keeping connected despite the problems that we have had. And we promise that we will upload the YouTube talk from Jose Manuel so that you can enjoy it later on. Patricia, if you want to say something.
No, thank you very much, and thank you for your patience. Yes, we have never had so many problems together, but thank you for staying with us. Okay.
Bye-bye for now.
Bye.